Hello, this video will be going through the study in the EMBO journal. Cancer is a common disease which has many different types of locations that it can impact on the human body. There are numerous factors that play a role in contributing towards the onset of cancer. Skin cancer is a very common type of cancer impacting many individuals. Environmental carcinogen contributes to 75 to 80% of diagnosed cases each year. UV radiation is an environmental carcinogen which is universally present in the form of sunlight. UV radiation has a high prevalence of somatic mutation which ultimately causes mutagenic DNA lesions. These mutagenic lesions are broken down into two main classes, CPD and 64PP. CPD is a more common type of mutagenic lesion which links UV radiation to DNA lesion as it is more likely to cause mutagenesis and malignancies. Mutagenesis and malignant lesions are formed from CPD and 64PP as they form covalent linkages from severe structural distortions in DNA and disrupt other cellular processes. This study looks at an area which has not been researched extensively, carcinogen susceptibility and mutation heterogeneity. It was hypothesized that the factor regulating localized genomic carcinogen susceptibility has extensive consequences in carcinogenesis by defining probabilities for mutation. The experimental procedure was executed by firstly treating the IMR90 cells. The DNA cells were exposed to UV radiation with 100 joules per meter squared for less than 10 seconds before the cells were lyased with 1% SDS buffer. Then, immunoprecipitation was performed by purifying sonicated single-stranded DNA with anti-CPD bodies. The cells were then repaired with photolyases. Slot blot analysis was performed. The cells were sequenced using Illumina sequencing kits. Genome and epigenomic analysis was done. The analysis was conducted by doing whole genome quantification, which was determined by pooling the DNA lesions. This graph shows box plots of the DNA lesion abundance at the top. Middle panel shows the mean of the histone modification and DNA's 1 hypersensitivity for chromatin states. The bottom panel shows the enrichment significance of genes and repetitive features. Lamin analysis. This shows the significance in enrichment. The top 5% of all UV susceptible regions and line are enriched with LADs, as opposed to regions outside of LAD or a non-LAD random control. Random forest regression. The first bar graph shows the 10-fold cross-correlation. The graph reveals 79% of carcinogen susceptibility. The graph on the right was also obtained with random forest regression. The red line is the abundance of UV lesions and the blue line is the C to T mutation frequency in the malignant melanoma cell. The line graph shows a strong correlation between DNA lesion accumulation and mutation in the C to T position on the chromosome analyzed. A whole 3D genome model was made using the Chrome 3D model of the IMR90 cells, capturing the positioning of TADs. Model on the left shows the mean UV lesion abundance against the mean radial position of each of the chromosome. The blue denoting the regression lines and the gray molecules denoting the standard error interval. The model on the left has the representation of each molecule as TADs. From these molecules, it can be seen that carcinogen susceptibility is a lot higher in TADs. Melanoma analysis was done by comparing the frequency of the melanoma mutation against the abundance of UV lesions. These graphs are consistent with showing that there is a correlation with the presence of UV lesion to C to T mutations in melanoma and in melanocytes. Therefore, the study concluded that the epigenome architecture in non-malignant cell does present to be a primary barrier to cancer development, that this is done by regulating susceptibility to carcinogen.